So, as previous speakers have already discussed about various omics, so I will not take that much time. Just a few points that all the omics data, whatever you will use, once you analyze the data, they are very like one of the thing is they are very high dimensional data. So, as this workshop is mainly focused on data analysis part, so you have to learn and apply new methods like the classical method which you use applying for applying the basic molecular biology data analysis that will not work most of the times. That's what we learn in various omics data and how it helps that as previous speakers have discussed, like basically you get from a system biology point of view, we get a holistic view. You can study one gene or one protein, but if you want to really know about a disease or any condition, actually we know that every protein and gene interact with each other, and many RNA regulate some gene express other protein, that protein may downregulate other protein also. So omics helps us in connecting those dots between the protein and genes. Next slide, please. So transcriptomics is basically the study of the transcriptome. So transcriptome is the whole RNA set inside a cell. Basically, all as you know, different types of RNA, as there are mRNA, miRNA, tRNA. Many different RNA are studied in transcriptomics. But basically, when you want to measure the gene expression, we go for mRNA. As you know, from a DNA, mRNA is produced, and from a mRNA, protein is produced. So if you want to study the gene expression, we go for mRNA. And there are two major techniques in transcriptomics. One is RNA-seq and another is microarray. As Dr. Arup has discussed about protein microarray in similar, and the like the microarray used in transcriptomics is very similar. Just in place of protein, we like use RNA. We make cDNA from RNA, and then we study the gene expression through those arrays. And other parts like the chips and all those things are very similar to protein microarrays. So next slide, please. So, basically, you can compare between microarray and RNA-seq. RNA-seq is a very new technique. Microarray is a little bit old. So there are several, basically, okay. So few points you have to remember that if you want to discover any novel transcript, then you have to use RNA-seq. But if you have the reference genome and like if you know the genome sequence of your organism on which you want to work, then you can go for microarray. Except this also, the dynamic range and sensitivity of RNA-seq is much higher. Dynamic range is basically if your gene is like how much fold upregulated or downregulated than another, like in control to disease, the fold like how many times it is overexpressed or underexpressed, that is much more sensitive in RNA-seq than microarray. And also the reproducibility in RNA-seq is in many cases it is like more. And if you okay, so other things are basically mm, yeah, so in intensity also, like the labor intensity is basically high in RNA-seq, but it gives us better result than microarray. So most of the transcriptomics world has slowly moved on to RNA-seq from microarray, except some very special cases which we can discuss later. Next slide, please. Okay. So these are the public databases, as previous speaker have discussed about the databases. If you don't have time or money to design your own experiment, you can just download the data and analyze it, and you can publish paper with them also. You just have to cite the authors. So these are some of the databases where you can download both the microarray and RNA-seq data. Omics.di is a umbrella databases from which you can download many different kinds of omics data, starting from genomics, transcriptomics, and all those things. So transcriptomics data also can be downloaded. NCBI Geo and NCBI like SRA, these are basically transcriptomics databases from which you can download the microarray and RNA-seq data. Also, there is Array Express from EMBL EBI from which you can download the data. So there are also many filters are available which we can show you in like a hands-on session later on. If you have a design in mind, like a particular organism, if you want to study human, if you want to study a particular disease, if you want to tissue type or cell type, whatever you design, you can just search in the databases. Okay, I want this organism with this disease and this type of cell or tissue. Then it will give you some results. Then you can just download the data and analyze in your own machine to get some novel results. Next slide, please. So this is basically just microarray, how microarray works. We can see, I already previous speaker have discussed about the array part, so I'll not go into details. So the right side is the analysis part, how it is done. Basically, the raw data, like microarray analysis is mostly done using R language, and you can import the raw data, download the raw data. You can use these are some of the packages, okay, which we don't have that much time to go into details. So basically, we different packages, bioconductor packages in R language to get from the raw data till the differentially expressed gene. Differentially expressed genes are those genes whose expression varies between a control or disease condition or between two any cell types or whatever study you design. Their difference you can make. You can just go through the pipeline in the R. Then you will get a list of genes which are differentially expressed. Then you can take forward those genes for further biological analysis. Next slide, please. So this is also the workflow of RNA-seq data. It is a little bit complicated than microarray. Microarray is a little bit straightforward because here you have to align the raw count first to the reference genome or transcriptome. Then you have to do the differential expression analysis on those raw count. You have to, in between, you have to first normalize it and do quality control and a lot of things which we will discuss. If you join for future hands-on session, we will discuss in details how to analyze the RNA-seq data. So next slide, please. So this is just Brainport, as Aparna has shown. Also, Brainport has a section about BDTM, which have the transcriptomics data. Now we have integrated the microarray data, and we will integrate RNA-seq data very soon. So you can just go to Google and search for Brainport and search for any particular gene you have in your mind, and you will get the expression of those in microarray studies. Many microarray studies, around 36 microarray studies, we have made the analysis and uploaded the data here. So you can get the expression of a gene, and you can like see if it matches with the literature or if it matches with your hypothesis or not, just in a like overall way. Then in order to do the analysis, you have to learn how to run it in a programming language. But I wish everyone can go to Brainport once and check the expression of different genes in different diseases. Okay, next slide, please. So these are some of the latest research, not that much latest, but still spatial transcriptomics is the method of the year in Nature in 2020. So what is this? One of these is single cell transcriptomics and another is spatial transcriptomics. So in single cell transcriptomics, we study one type of cell. Like tissue consists of different kinds of cell. So there are cell heterogeneity inside your tissue. So when you do bulk transcriptomics, we cannot capture those heterogeneity. So in order to capture that heterogeneity, we do single cell transcriptomics, and it has many applications which we cannot go now. Okay, so then the another is spatial transcriptomics. This is relatively new, and it is being used highly now to create the map of different tissue. Like you can use spatial transcriptomics to get the map of a whole tissue, like a whole brain or any organ. You can get, okay, in this tissue at this like cell types are there, and this cell types, this genes are expressed. So you can just basically create a map of any organ and use that for further research. Next slide, please. Okay, so as I discussed, in the future workshop, we will learn about how to analyze a microarray data, starting from the downloading the raw data till we get the differentially expressed gene. we'll learn about RNA-seq analysis workflow also, and also how to basically download the data from the public databases and load it into a programming language environment, and how to get some useful genes for that disease. And also we'll learn how to, once we get the differential express gene, how to do some functional analysis like using Reactome and other KEGG and other pathway analysis tool, how to make some biological sense out of those top genes. Thank you very much. If you have any question, I think we can take, or I can answer your question in chat also if there is no time.